Hello and welcome back. The topic for this video is going to be mutations, okay? And whenever I bring up the term mutation in my face-to-face class, usually the first thing that comes to mind for students are, something abnormal, something weird. The frog with, 5 legs, snake with 2 heads, or if you're really cool, you think about the X-Men. Okay, which in that case is mutations that are more beneficial, all right? But with mutations, that's something to keep in mind, okay? Mutations can be either detrimental or beneficial, all right? So they can be bad or they can be good, like in the case of the X-Men, all right? But really what we're talking about with mutations, what a mutation is a change to the DNA sequence. And what happens, all right, when you alter the nucleotides, is that can have an effect downstream on our protein, okay? When you change that DNA sequence, that's going to result in an RNA sequence that's changed, and that RNA sequence may change our protein because it can put in a different amino acid, okay? So mutations alter DNA sequences. They can essentially change the genes that are present within a particular cell, okay? And with those mutations, all right, we've kind of got 2 classifications that they can fall into. They'll either be what's referred to as a point mutation. A point mutation changes just one nucleotide. Getting ahead of myself again. Changes one nucleotide, okay? And then you can have rearrangements. In the case of rearrangements, you kind of get large chunks of DNA that can either be deleted or can be inserted somewhere else on the chromosome, okay? And that can result in changes as well. These tend to be bigger changes. These tend to be not quite as big, all right? But what I do want you to be able to identify are the different types of mutations and the change in the DNA sequence that's associated with that particular mutation, okay? So we've got a nonsense mutation, okay? And like that says, nonsense, it's going to make something that doesn't make sense, okay? In this case, a nonsense mutation, I want to say inserts, I want to say change. It's going to change our codon to a stop codon, okay? So you get a mutation that happens that changes what would normally be an amino acid to a stop codon instead, okay? And what that results in is what we call a truncated, which is a longer word for short, protein. And usually, these are going to be non-functional. All right. at best, they may have an altered function, okay? But it's not going to work the way it's supposed to, all right? And in the PowerPoints, I've got a diagram of what happens, what the mutation is that changes the DNA sequence, which gives a resulting change in the RNA sequence to change the protein, okay? So we've got the nonsense mutation. We've got what's known as a silent mutation, okay? And this is what can happen when we're talking about the wobble. Remember that third base in our codon? Silent mutations, very often it's that third base that gets mutated. And what happens is you have a change to the DNA sequence, but, and we've got a big old but in here, the same amino acid is used. Okay, so it's not changing the amino acid that's inserted into the protein itself. So that's what's known as a silent mutation, because you don't really have any physical trait associated with that mutation. There's a change in the DNA sequence, so something happened, but it's like mutating our proline that's a CCC to a CCA. Either one of these is going to insert our proline as the amino acid. So the protein's still going to function absolutely perfectly normally, okay? So we've got the nonsense, we've got the silent, and then we've got the missense mutation, okay? Missense mutations change the codon to insert a different amino acid, okay? And the book likes to use hemoglobin as a great example of this, because people who suffer from sickle cell anemia, believe it or not, the change in their genetic code is just one DNA nucleotide that's different from normal. And what happens is that results in the normal glutamate. Instead, the amino acid that's inserted into that hemoglobin protein is a valine, okay? So hemoglobin has just, hemoglobin. People who have sickle cell anemia have just one single amino acid difference in their hemoglobin versus normal, okay? Changing out that glutamate for valine results in a protein that doesn't work quite the way it's supposed to, okay? So that's our missense mutation. Missense meaning we're putting in a different amino acid. You're not putting in a stop codon though, okay? The protein will still keep being made, all right? Missense mutations can be functional. They can be sort of functional, like with hemoglobin, or you can change one amino acid and have the protein be completely non-functional, okay? So again, you've got a change at the DNA sequence, results in a change in the amino acid that's inserted, okay? The next type of mutation that I'm going to talk about, because I've run out of board space here, I'm going to do that in the next video. The next one I'll talk about is going to be frame shift mutations, okay? So if you have any question about the nonsense, silent, or missense, please let me know. Otherwise, next video we'll talk about what happens with a frame shift. Thank you for your attention.